brief introduction of what gene expression is and the different technologies that are used to quantify the abundance of mRNA in tissues. And then I'll talk about data normalization and the distributions of gene expression that we encounter and why we want to kind of deal with the differences in these distributions. So there's a couple of ways people are doing this. And then I'll briefly touch on differential gene expression, but just like a little bit, and also in the context of kind of batch effects. So I hope it's somewhat useful, and it's like mainly pictures, and so you get like an intuition of the data. So gene expression is very interesting for studying mechanisms in biology, because how cells kind of regulate the internal functioning is by controlling the concentrations of proteins in a given cell. And so this is completely regulatory because every cell obviously is genetically identical, right? So the only difference between a liver cell, let's say a kidney cell, is which genes are locked down and which genes are producing mRNA, which is then in turn turned into protein. So, a quick course in genetics. So you have DNA, and the DNA is kind of like a blueprint template from which through RNA polymerase, you go from DNA to RNA. So this is kind of, just a single-stranded sequence. And then that gets translated into an amino acid chain, which then folds itself, or like with help of other proteins, into a three-dimensional structure, which is like the protein. And the protein has the chemical properties, that really regulate chemical concentrations within a cell. So if we look at on the right, you kind of see these two strands. So one is the primary RNA transcript. So this would be the exact copy coming from the RNA polymerase readout. So here you have exons and introns. And so eukaryotic cells, like human cells, they're like different than bacterial cells in the sense that they can recombine the original DNA sequence into different types of proteins. So as an effect, there's actually bacteria that has significantly more genes than the human DNA has. But eukaryotic cells and human cells have the ability to produce many more proteins because they have something called splicing. So what happens in splicing is so first of all, they always cut out the introns, so they're never part of the mature mRNA. But then the exons, you can imagine as sequences that have very specific functions and domains. And you can kind of mix and match those exons. So you might have a mature mRNA from this primary RNA by maybe only taking exon 1, or you could take only exon 1 and exon 3, or you could take all three, right? So there's a lot of variations that you can have. So this is a completely separate step, which is quite complicated. And I don't go into like too much detail, but like what you can remember is that you can analyze biology always either on a transcript level, and the transcript level, you would always measure the mature mRNA, so you can actually see differences in the splicing variation of a gene. Or you can have like a lower resolution where you look at a gene level. So you would kind of collapse all the transcripts that come from some primary RNA sequence and associate the counts that you found to that gene. And in general, I'm not sure what the average is. I think it's like every gene has on average 8 different transcripts. But some can have many more, and some only have one variation. So there's a lot of complexity there. So if we work with this gene level, we kind of ignore that complexity. So there's there's a couple of major technologies that are used to kind of capture this whole genome-wide gene expression. And initially, the technology that came out was the Affymetrix microarray, like microarrays in general. And so this kind of technology, this like whole genome-wide technology, was made possible initially when the human genome was first sequenced. So before the year 2000, people really didn't know how the human genome looks like at all. And so all these technologies rely on our understanding of the sequences of the genes. So, when they finally sequenced the genome, and then they realized, oh, there's only 20,000 genes, how is that possible? Because, like you could have asked big professors or like famous researchers like 2 years prior, and they would have told you there must be at least 100,000 genes. So this was a pretty big surprise. So the Affymetrix chip then came, and it was really the first technology that could measure all genes, or nearly all genes, in parallel at once. And that was a huge technological breakthrough, because before you could only really measure individual genes, or like a panel of 10 genes and how they change in expression. So this was pretty big. And like even in 2010, people didn't really believe that that is giving any reasonable insight. So it was fairly contentious for a couple of years when it came out. But now this is like the main way people quantify the activity of genes. So the Affymetrix chip, you can kind of see there's like this small square. So it's like a pretty small kind of square platform. And then with like incredibly tightly packed sequences, they have these probes. And these probes, they're usually 25 base pairs long. And they're complementary to the RNA that you want to capture. So by knowing the sequences of let's say, your reference genome, like your, let's say, human genome, you can put these probes into very specific locations, right? So there's, it depends. So in like some platforms, this one might have like 500,000 cells. And then each cell has one type of probe in it. And so now you can wash RNA that you extracted from tissue over this chip, and they will attach to the matching probes. And because they're labeled with fluorescent tags, you can then look you can excite them with a laser, and you can read out the positions that glow on the array. And that will tell you which probes identified fragments from the sample that you had. So sounds like pretty crazy technology, but this was the first they came up with. And so as an example, so usually when you, so how these work is you have these probe sets. And a probe set is usually an overlapping sequence of probes from one specific location on the genome. So this is for mouse, like an example. And we have this probe set ID up here. It's like 1415771 AT. And so it stretches a whole sequence of about 129 base pairs. So what they do is, so this is actually a picture of a chip and how the excitement of the fluorescent tags can look like. And then in red, you kind of see the position of the probes for a single probe set. So they're kind of randomly distributed over the surface of the chip. And that's important because you do tend to get weird artifacts. So you can kind of see some banding. Sometimes you will get lines that are affected. So by randomly placing all these probes into different areas, you can control for those effects. So in the end, if you want the intensity of a gene, you would have to average out the intensity of the probe set. Okay, so if you ever encounter the Affymetrix data, you will not have intensities by gene, but you have these probe set intensities, and these probe sets are usually some numeric value, underscore, and then some suffix. And so those are kind of meaningful. Took me a long time to realize that they actually have a meaning, but they kind of specify how unique they map the reference genome. So you have AT if all these probes in the probe set really are very unique to the location that they're supposed to map to. But then you have also probes that are like less good, meaning they can cross-hybridize. So when does this happen? So there's certain gene families where the genes will, by their sequence, look extremely similar to each other, right? So like, for example, all your smell receptors, so those are just, it's a large family of genes, let's say 600 genes. And sequence-wise, they're extremely similar, except for like tiny differences. They make up their specificity for like certain chemicals. So those are harder to measure with Affymetrix probes because of this cross-hybridization. So this way, it's not always clear if you get some intensity score from a probe set, if it is related to one gene or the other. Okay, so then, so this is one of the major technologies that existed. Then I add this because some people are working with this data set. So this is a little bit more exotic, I would say. So this is not like a mainstream technology that people are using. But so this is the large-scale expression analysis of links, which is the L1000 technology. And so this technology works by, instead of like having this array where you have, you know, like a kind of like a checkerboard and you have like areas that are coded with a certain probe, you have these tiny beads. And so these beads are coded with a unique probe, like similar to the Affymetrix chip. And these beads, they come in 500 different colors. And this is 500 is a technical limitation where the laser can only differentiate these 500 different colors. So this would limit you to 500 probes, or to be more accurate, it would limit you to measuring 500 genes. So they use a trick. And okay, so like just briefly how this works is they mix the beads with the probes and the mRNA they extracted from the samples, and then they run them through a laser. And the laser can detect the color of, or like they have a detector that can measure the color of the bead, so they know what gene we're looking at. And then they measure the intensity of the fluorescence of the mRNA fragments that bound to the probes. And that gives you the abundance of the mRNA in the sample. And then, you know, because the people were like not happy with just measuring 500 genes, they want to measure 1,000 genes. And how they do it is they overload the color of the bead with a second type of probe. So that means a single color of a bead can come from two separate, can be coated with two different probes. And you don't know that. If you just look at the color, you will not be able to know which, you know, which probe it is. But, so this is kind of this pipeline. So the whole point of this technology is that you can automate this with robots. So you have this plate, and it has 16 by 24 wells in it, meaning you can do 384 experiments in parallel. And you don't need humans to like do any experimentation. It's all like robotic. So if you go to the Broad Institute, you can kind of see machines just like loading those. And very fast lab techs that like just churn out a lot of stuff too. So, but so you have these wells, and then once you run it through the program, you get these double peaks for each color. And so this is the trick of going from 500 to 1,000 genes. And so they do this by let's say if we have a single color, right? We have these red beads, and they can be coded with probe 1 or with probe 2. They can change the ratio of how much probe 2 and how much probe 1 was used. And so they expect to get a bimodal distribution, and like one distribution is smaller than the other because they know the ratios of the probes, how they mix them. So by just looking at these peaks, they can build some Gaussian mixture model, or I think they use a k-means in their code to identify which probe belongs to which peak. And so this is the intensity that they get. And then they go on and do their normal normalization. There's a lot of stuff they can do. And then from the 1,000 genes, they have a linear model. I think now they changed it into like some prediction model that predicts the gene expression of 12,000 genes that were not measured by extrapolating it from the 1,000 genes that were measured. And then some people, might be familiar with this, where you kind of take all the gene expression, you try to get these signatures. So I'm not going into that. And then so the most predominant technology that's used nowadays is called RNA-seq. And RNA-seq is quite similar at the beginning if you build your sequence library. So you extract mRNA from tissue that you're interested in. And then you break it up by a process called sonication, which basically just you put a high frequency, and like then the mRNA starts to vibrate at the same frequency. And depending on the frequency, it breaks into fragments of approximately the same length, right? So often they try to break mRNA into fragments of about 200 base pairs. So when they have that, there's like, some more chemistry involved. But in the end, they will run it through a machine that can read the fragments. I haven't like shown exactly how this works, but so in this case, they literally just rebuild these sequences, and they can just read, like for all the fragments that they found, what the sequence is. So then in the next step, they will just have to look at the fragments that they found and identify on the reference genome where it was from. And so this is the alignment step of the process. And then for every fragment that was aligned to, let's say, a certain position where a gene is annotated on, that gene will get a read count associated to it. And there is two variations of this. There's single end and paired end. So single end is really if you have this, sequential read, and you only like sequence it from one direction, and you associate it to a reference genome, and that's it. So pretty simple. And then paired end read is where you have you kind of sequence you sequence it from both ends, and you know that they're matching. So then you have some distance in between it, and it has some advantages, especially if you're looking for indels, like genetic variations where like, a mutation occurred where like new nucleotides were inserted into a gene or something like that. But this can also be used for mRNA. And it's supposed to be just slightly more error tolerant because you have two reads. It's more accurate and like probably less prone to like misalignment or like mismapping on the reference, so especially for genes that are very similar on their sequence. So the raw data that you get from RNA-seq looks something like this. So it comes in these fastq files, and so there's so fastq, the Q stands for the quality score, which means you have these weird. So in this case, it's like I don't even know what the word for that symbol is. It's just like that that vertical stripe, right? So the first line is just the read ID. And then the second line is the nucleotide sequence that was detected by the sequencer. And then again, you have the ID. I don't know why that is double. I have never figured that out. And then the fourth line is the quality or the Phred scores of the individual reads. And in general, because it's kind of an iterative process, if you sequence the mRNA, the further you go along, the more likely you are that there's a slip up or some error. So oftentimes the quality degenerates the longer you sequence. But so in this case, so how this encoding works is there's usually, a range of quality scores, and it's really dependent on the technology of the, the sequencing technology that was used. But in general, it gives you some quality of how likely it is that the read is accurate. And so like in this table, you can kind of see it's like on a logarithmic scale. Or like it's kind of like exponential. So, if the FAT score is very high, so this would be this encoding here at the beginning. So it's very high. So it's about, it's pretty much, it will be accurate. And then later on, these are somewhat wrong. And so these files can be pretty large, right? Because it's all text, and you can have millions of reads. So they usually, like, each of these files is about, 4 gigabytes or something, or it can be 30 gigabytes. And so it really depends how many reads you actually want to sequence. So this is like a cost question, right? So you can have extremely high coverage, but you will have to pay. So because the sequencing, you literally pay by how many reads you sequence. So this is kind of if you want to plot the quality per base. So imagine, so in this experiment, the sequencing length was 35 base pairs. And then you kind of see the score distribution, or the FRET score distribution, from like the first position to the last. So you can kind of see that it falls off in the end. So the most modern aligners, they can deal with this stuff. So it's something that you don't really manually ever have to worry about, but it's good to know that it exists. Okay, so the main reason why RNA-seq kind of beat Affymetrix chips, I mean, there's a couple of reasons. I think most relevant is that it has a higher dynamic range, so it's just more accurate across the full spectrum of gene expression that you encounter. So Affymetrix chips, I think, have issues, especially for genes that have fairly low expression. And RNA-seq is just more accurate. And you can kind of see how over the years, the number of published studies for this whole genome expression really shifted from these Affymetrix chips, or like these chip-based methods, to RNA-seq, right? So, and this is still going exponential at this time, right? So there's hundreds of thousands of RNA-seq experiments, but there's still people that do Affymetrix chips, so it's still out there. So that's why I added it to the slides. Okay, so the distribution of gene expression. So I wanted to put it here so you have seen it. For one, so on the left, you just see a simple box plot of the gene counts if they are log transformed. And this is just for 30 random human gene expression samples that are publicly available. So I extracted them from ArchS4. And so you can immediately see that some experiments, for example, 22, has fairly high log2 gene counts, while other samples like sample 5 has extremely low log counts. And so on the right, you see I pulled out 2 samples from the left. So it's sample 1 and sample 9. And what you see is there's a peak close to 0. And then there's a second peak, right? Very good. Peak. So what this means is that approximately half, it's close to half of the genes are usually not expressed in a given tissue. So that's kind of important, right? So the way how a tissue is maintained is by locking down genes, usually through something called methylation marks. So they can't really be expressed, they can't produce mRNA, because the DNA is not accessible to the regulatory mechanisms to actually read out the mRNA. But what's like the point that I want to make with this slide is that so we have these bimodal distributions, but they actually are not really that comparable, right? So this is for like all the genes, right? So it's just plotting distribution of the scores for the genes, but you see that that sample 9 has on average much higher expression than the sample. Wait, did I change the? Sorry, I mixed the colors around, whatever. So sample 1 has much lower expression than sample 2. So that is a problem for a lot of the downstream analysis that we're going to do, right? So imagine I want to know if a gene, let's say I'm interested in FOXM1, it's some random gene, and I want to know is it expressed higher in sample 1 or sample 9? That might be a bad question to ask, right? Because just how the depth of the reading went, right? And because they came from 2 different experiments, maybe the one group spent more money in just getting more reads. So that's why they get higher expression. But that's not that meaningful, right? So I have to correct for those differences. Okay, so these are common ones that are used to correct RNA-seq data. And so let's talk about the first 2. This is like RPKM and FPKM. And they're virtually the same thing. One is applying to single-end reads and the other one to paired-end reads. That's why I put that slide in, so you kind of, you know, see the same thing. So this is literally just how you deal with, it's just nomenclature. So okay, so FPKM or like RPKM is reads per kilobase of transcript per million mapped reads. So what's the logic behind that? So the people who came up with this method, they realized that genes come in very different lengths. Okay, so there's a gene called Titan, and I'm not 100% sure how long it is, but I think it has like 100,000 base pairs or crazy. It's like extremely large. And so if you look for mRNA, because you fish out mRNA from this pool of genes, the genes that are extremely long, they will have much higher chance of contributing like sequences in your read file. So this method tries to correct for the length of the transcript. So here the length of the transcript is LI for like transcript I, and then we have XI. So this is to be accurate, so this is still all on a transcript level. So this would be the transcript counts, like the counts that are associated to a transcript. So what we basically want to do is we normalize, we divide the number of reads by the length of the transcript, and then we also normalize it by the total number of reads that we have in the file. So in the end, you get this term xi over li times n times 10 to the power of 9. And what's important about this is if you apply this normalization for different samples for every gene, the samples will still not add up to the same value, right? Because, is that correct? Yeah, pretty sure. So now let's keep going. So the counts per million map reads is a simpler version. So this one doesn't account for the length of the transcript. It just accounts for the number of reads in a file. So if we apply this normalization, we should get, to like a more comparable comparison between the plots we had here, right? Okay, and then like one of the more popular ones is TPM. So they're all kind of related to each other. TPM is kind of a mixture of CPM and RPKM. And so this one also corrects for the transcript length. But so if you apply this normalization, if you sum up all the reads from a sample if you sum up all the TPM scores for a sample, the sum is 1 million, right? Because if you look at this term below, you kind of normalize by all the scores over all transcripts, right? So as a sum, it will kind of just like cancel out, and you are left with 10 to the power of 6. So one thing, like as a remark, generally, if we look at something like differential gene expression, where we ask, is a gene higher expressed in one tissue over the other, normalizing by the transcript length or the gene length is really not that helpful, because that's actually invariant between the two states. So I'm personally not like a huge fan of these normalizations, but like a lot of people use them and they think they're important. But like realistically, like if you use differential gene expression algorithms, they do not want these kinds of normalizations. So they only rely on the raw gene counts, and they do their own normalizations. And I have an example of that coming too. Okay, so first, this is a method that I like a lot, and this is quantile normalization. And so here I have an example of 3 distributions, and they're just shown in like 2 different ways. One is just a density plot, and then the other one is a box plot. And one thing to note is that this density 3 is kind of like a bimodal distribution, while the other 2 are really just normal distributions. And so each of those has 1 million random data points that are used. But as I said before, density 3 are just built by mixing 2 normal distributions. So they have kind of different properties. Okay, so quantile normalization. So the purpose of quantile normalization is to force all these 3 distributions into the same distribution. So if we're done after this, so I'll show like how we do this, but basically if we would apply quantile normalization on those 3 distributions, we end up with this black outline of a new distribution, and all the values in these 3 distributions would fall into that distribution. So then if you look at the box plot, after we would apply quantile normalization, this is how the box plots would look like for those 3 distributions. And you can kind of see that because we force them into like an intermediary density, you can still see that slight hump on the left side. So we do change the shape of the distribution. Okay, so this is like a really simple example where the columns are our samples, right? So these are our gene expression profiles. And then each row represents a gene. So here they're color coded, so it's easier to understand what's going on. But so first we rank the values based on their expression, or we rank the genes based on their expression in the samples. And we keep the color so we can keep track of where the things went. If there's some like numeric error, that might be, I think I had this presentation like a while ago and then somebody found an error, so don't get too hung up if there's something that doesn't quite add up. So, okay, so once we did the re-ranking, we replace the values. So for example, for the values that are in the first row, we replace it with the average of that value. So in this case, for the first row, it's the average is 3. So like 2 plus 3 plus 4 divided by 3, right, is 3. And so we do this for all the rows. And then once we do that, we can change them back to the original position, which is easy because we color coded them. So we can like just do that. And so now that we reordered the vectors, this is our quantile normalized expression. And to no surprise, because we replaced the rows all together, right? So like all the values in the first row got replaced by 3, and then all the next ones by 5. The distribution of these values is now identical in all the 3 samples. And so this is what we wanted to do. And if you have something like gene expression, if I go back, so what I like to do with this data is, if I have, well, like let's go here. So if I have the data that I have on the right, so this is log transformed, and so I generally prefer to do a log 2 plus 1 transformation on the gene counts first, and then apply quantile normalization. This is mainly because it deals a little bit better with outliers. Because if we calculate the average across these rows, in gene expression, there can be some gene that has like 2 million reads for some reason. So in log space, it's not such a big deal. Okay. So another thing that's kind of important, and this is mainly used in algorithms that use differential gene expression, is that the algorithms are oftentimes skewed. So differential gene expression is oftentimes skewed by the absolute expression. So like an MA plot has 2 axes. The x-axis is the average expression of a gene across the samples that we look at. And then the y-axis is the dispersion or the standard deviation of the gene expression in those samples. And so this is kind of a simple example of that. And so one of the issues is that oftentimes in biological data, the dispersion is somehow has some mutual information with the average expression. So like a simple example that like would kind of explain that is that we would expect to have potentially higher random noise for genes that have very low expression. And this is mainly because if we have, let's say, a gene that has 100,000 reads, if there's some technical noise that will add, plus minus 100 reads, that gene, the log in the log scale, that like doesn't really matter, right? We would not really detect that as like noise between the samples. But if a gene has only an expression of, let's say, 200 reads, and we have some technical noise that adds plus minus 100 reads, that is a huge amount of variance for that value. So but in practice, so you get these MA plots. They are produced often by these differential gene expression studies, and they will just show like each point here is a gene, and it's the average expression in log scale, and then some dispersion score explaining like often it's the fold change between 2 types of experiments, something like that. So we kind of want to correct for that, right? So the idea is that there shouldn't really be a bias between the average expression and the dispersion that we measure between, or like the fold change that we detect between those samples. And yeah, so more or less I talked about that. So there's many ways to model this. But so important to notice is that this affects genes with very, like low expression. So they can end up having higher dispersion and also short genes, right? So as I mentioned before, some genes have only a couple of hundred base pairs and other genes have 100,000 base pairs. So the gene that has 100,000 base pairs will always have a lot of reads in the read file, so it's not as prone to that noise, right? So we kind of want to correct for that. So the way to do this is like Lowess or Loess correction, which is basically following like a locally weighted scatterplot smoothing. That's, I guess, the thing. And like the way you can fit these models, there's like many ways. But in practice, so here's an example function, which is quite simple. It's just like a value like 1 to 10,000, and then I just have an offset for the x and y, and I just plot the MA plot as described with the log x1 divided by x2, and then for the average, it's just average of the x1 and x2. So this is just this line, so this is the truth, and now I can just add some random noise, and then I get the scatter plot. So now if I want to re-find the truth, I can just have this local smoothing where I just, fit a line at like these given windows, and it pretty much gives me this function. And so once I found this function, I can basically just straighten out the data by, imagine, just bending that black line straight, right? And then normalizing everything accordingly, like all the blue points I could kind of correct for that line, and that's how we can do that. Okay, so from so I think I mentioned like some of it, but basically when we want to get from like raw data, which is this like from these FastQ files to differential gene expression, there's like a couple of steps. So the first one is we want to do some quality control, which I've shown. Then there's this read alignment step. And there's a bunch of algorithms. The most popular one is called STAR. But there's other ones that are like faster. So like Callisto, for example, is more lightweight. And then so then you can associate these transcript reads to genes by using these genomic annotations. So once you found the position of a read on the reference genome, you just have to count up how many reads overlap with regions of genes. Then you can normalize your data. And that really depends if you want to normalize. So depending on what differential gene expression algorithm you want to use, they don't want you to normalize anything because they do everything like internally. But so for example, if you want to do something like correlation between genes, then normalization is important. Because if I just jump back real quick. So if we think of this on the left, and we can see, let's say we want to take a single gene and then calculate the correlation to another gene across those samples. If we would have nicely behaved data, and the genes are not really, have nothing really to do with each other, then the correlation should be 0, right? So if I would calculate all pairwise gene correlations on this data, how it is right now, and it's not normalized, I would get a lot of positive correlation, right? So just because they have a lot of shared variance, just because of the sample count distributions, right? So we want to get rid of that. So that's something that quantile normalization would be good at. So if this data would be quantile normalized and we calculate all pairwise correlations, the correlations should center around 0. Okay. Okay. So now I'll just briefly talk about differential gene expression analysis. So this is the, I guess, an important step if you want to identify genes that are different in one set of samples versus another set of samples. So this could be a healthy tissue versus a cancer tissue, right? And so as I mentioned before, like most of these differential gene expression algorithms are not using any of the normalization methods that we talked about before. So they do their own one. So I'll just show the DGE method they do, and then you can kind of, think about if it makes sense or if quantile normalization is better. But so in this case, this is one of the more popular algorithms, and this is what it does. So first, again, we have this problem of the differences in library size. So you can see that sample 1 and sample 2 have very different total number of reads, right? So sample 1 has half the reads than sample 2. And this is the library size problem. And then the library composition is what we're actually interested in, right? So in this case, A1BG, for example, the first gene, has much lower expression in the first sample than in the second sample. And then A2M has a much higher expression in sample 1 versus sample 2. So we generally want to correct for the library size, but in some instances you do have to worry a little bit about the library composition. Okay. Yeah, so this is a very common library, so I'll like skip that. And there's a couple, so there's like DGE HR is another popular one. So I just show the normalization that DGE does, so you get an idea like of other ways of dealing with this. So here we have a toy example of 3 samples with 3 genes. And then we have the gene counts in here. So what we want to do is we want to scale the values of each of the rows so they're comparable, right? So just by like immediately looking at sample 1, 2, and 3, we see that sample 3 has a lot more counts than the other 2. So, okay, so let's go. So what we do in the first step is we take the log, and this is for the same reason that we take a log before we do the quantile normalization. So it's kind of to deal with outliers. And because there was a 0, we already have a negative infinity, right? So that's why usually when I do a normalization, I do the log 2 plus 1 thing. So then a 0 is a 0. And okay, so we have that, and now we calculate the average for each row. So this gives us the average log expression, right, per gene. And because we can't really do much with negative infinities, we remove that from the further analysis. And I guess that makes sense. So like you don't really want samples that have 0 expression in your analysis, right? So imagine a gene is 0 in all experiments. It's not really that interesting. Problem is if you have, let's say you want to apply this on a very diverse large data set, that might be tricky, right? Because a gene might be in one like random tissue, it might not be expressed, so then it's completely removed from the analysis. But so this is what it does. So it, tries to get rid of any gene that has a 0 in any of the samples. So in this case, we're left with 2 genes. Okay, so now subtract the average log, right? So we have the we have our genes in log space on the left. We have our geometric means of the logs. So now we center the rows around 0. So this is what we get. And so from this, so now from these centered genes, we can calculate a median for these log values that we found. And so median is also, again, to make it more stable against outliers. And this is now our final scaling factor, because it kind of tells us how much we have to correct sample 1, 2, and 3. And because we calculated everything in log space, we have to first bring them back to, I guess, the normal world. So we have the exponent of those. So now we have the scaling factor 0.4, 0.7, and 2.5. And so the scaling factor, we actually divide our values. So if these were so we had our original values in this top table. So now with the scaling factors that we identified, we divide these values with their corresponding scaling. So a 2 turns into a 5, and then like a 33 turns into like an 83 because it's divided by 0.4. And then sample 3 we divide by 2.5. So these values get smaller. And as we saw before, this is kind of what we wanted, because we already saw that sample 1 and sample 2 have fewer counts than sample 3. What's to note is that because we didn't look at gene 1, right, this could have been an issue. Let's say if sample 2 had 1,000 counts on gene 1, we would have never taken that into account when we calculated the scaling factor. But then again, this is only a toy example. So if you have thousands of genes, in practice, it's probably not a big deal. But that's it. So these were the scaling factors. And then with these normalized counts, it would then try to calculate differential gene expression. So this was DGE-2 normalization. So differential gene expression, quite simply speaking, is we look at the distribution of expression in one group and then compare it to another group of samples. And we call a gene differentially expressed if these two distributions are statistically different. And so there's a couple of assumptions, and every algorithm for differential gene expression kind of takes different assumptions into account. So for example, what is actually the distribution of gene expression, right? So in this plot, it looks like it's a normal distribution, but some algorithms will tell you that it's actually a negative binomial distribution. So they vary in details how they approach this, but in general, there's an algorithm called Lima-Voom that just assumes that the data is kind of normally distributed, and it was developed for the Affymetrix chips, and it still works pretty well. But it's something to consider. So one thing I wanted to add here is what is actually the number of replicates that you need, right? So if we look at this one, this is pretty luxurious, because just looking at the histogram, that means each group had, 30 replicates, 40 replicates or something. So that never happens, because every replicate costs a lot of money, and often it's a mouse, so you can't really do that. So biologists gravitate towards 3 samples per replicate. So you already see there isn't like a lot of leeway of noise, right? So if you would have a lot of noisy data with 3 replicates, it would be pretty difficult to show that there's a significant differences in the distribution, produced from like 3 values. So generally speaking, you would want more replicates. And so there's a nice study, and it's comparing all these different algorithms, and they ask how many replicates do you actually need to reliably detect differentially expressed genes, and it's 6 is the answer. But of course, no biologist will be willing to do 6 experiments, so it's a little bit of a dream. But so here we have an example of the true positive rate that we get. So the good news is, so each bar here is a different algorithm for differential gene expression. And on the left, you have the false positive rate, right? So this means, do I detect a lot of things that are wrong? And the good news is, so this is generally quite low. And but what we see is if we have only 3 replicates per group, so this is this first block on the top, we lose a lot of true positives, right? So a lot of genes that are differentially expressed, we can't really detect those with 3 replicates. And then that improves quite a bit until we get to 20 replicates where we can pretty much detect them to like 90%. But so the authors claim that 6 is kind of desirable. Yeah. So this is this is N. Well, you can always look at the paper. I uploaded the slides if you want, but so they kind of really point out that there is a real advantage of having more replicates. So if you ever have a say in like the design of an experiment, you can always reference this. Okay, so this is a little bit something that goes into a batch effect that people will observe. And a lot of these algorithms can deal with this, so they have built-in ways to control for those variables. So let's say here we have a very simple experiment. We measure a single gene, and we measure the expression of the gene in a control mouse and in a mutant mouse. And we have 2 labs. And they measure the gene expression 3 times for the control and 3 times for the mutant. So in total, there's 12 experiments. So on the left is just like some R code. But what this does is it kind of explains. So if we have this model matrix is called a design matrix. So this is something that these algorithms will take in to understand the experimental design, right? So you can kind of see that from 1 to 12, this is our samples. And then we see, okay, so the first 6 samples are from lab A, and then the other 6 are from lab B. And then we have mutants, which are the samples 4 to 6 and 10 to 12. And so if you look at this formula on the top, so this is kind of this regression model that we want to use to identify what variable is actually meaningful in predicting the gene expression. So this is kind of how Lima does it. So imagine we want to know the gene expression. Would it be helpful to know the mutant state of a sample, right? And it would be, right? So because we can see that the gene expression is higher in the mutant than in the control. But we can also see that it's quite important in which lab the measurements were taken, right? But we're not interested in that specifically, right? So by running a regression using this full matrix, so if I go in here, so this would be the result I would get if I run like a linear model in R. And output in different languages might look slightly different. So like all the way on the bottom, you see a p-value, right? And so we have a significant p-value, right? It's like 0.007. So this model that we have here, which is expression, and we predict that by using the lab and the type, and also the intercept. So we can predict the gene expression really well, right? This is why this p-value down here is very significant. But this is not the p-value that we're actually interested in, right? So what we're interested in is if the mutant type is important for the gene expression, irrespective of the lab, right? So, and this is this p-value that we have here with the type mutant. It's like the 0.009 value. So this is the p-value that we're actually interested in. So this value tells us how much more our prediction improves if we add the information of the mutant type to the model, right? So in this case, we would only use the prediction of the gene expression by using the, the intercept and the lab information. So this would get us some, significant information, but then by adding the mutant type, we get a much better prediction of the gene expression. Right? So this way, if you write differential gene expressions as such a regression problem, you can identify which variables are important. In this case, what we are really interested in is the mutant type. But you can also have, more than one state of mutant, right? So you could have control mutant and mutant negative or something like this. So there might be different types of states. Yeah, so that's it. As I said before, so you could, of course, also try to remove these effects of the batches. So like, let's say lab 1, like lab A and lab B, you can call a batch. You could, before you start doing differential gene expression analysis, try to remove differences between the labs. So you want minimal variation between them. But yeah, that's like kind of like a different thing. So there's algorithms called like batch removal algorithms. But yeah, I don't think there wasn't enough time to talk about that in detail. But okay, so I hope that was somewhat interesting. I know probably some people already read up a lot on gene expression, but just to give you like a nice like brief introduction.